Speaker will be Professor Sergio Guerrero from Instituto of Agrobiotechnology, if I translate well, in Argentina.
Hello.
Hello.
I think Sengot has my presentation.
Shall I? Do you want me to share it?
Yes.
Okay. So I'm just gonna.
Thank you.
Okay.
Good afternoon, everyone. I would like first to thank the organizers for inviting me to this international meeting. I belong to the Molecular Enzymology Lab of the Agrobiotecnologia Institute in CONICET and Universidad Nacional del Litoral in Santa Fe, Argentina. For several years, I have worked studying the redox metabolism in trypanosomatids, characterizing putative molecular targets for rational design of drugs. More recently, we started searching the carbohydrate metabolism in Euglena gracilis. Our present interest is the characterization of enzymes catalyzing the synthesis and degradation of paramylon. The first target we worked with was the UDP glucose pyrophosphorylase, an enzyme catalyzing the synthesis of UDP glucose, a critical substrate for paramylon synthesis. Here I want to thank Dr. Ellis O'Neill and Robert Field for sharing with us information of the transcriptomic research. With this information, we cloned the gene coding for the UDP glucose pyrophosphorylase and obtained the recombinant protein. We were able to perform a kinetic characterization of the enzyme, being glucose 1-phosphate and UTP, the substrates with which the enzyme worked more efficiently. Galactose 1-phosphate and TTP were also possible substrate for the enzyme, but it worked with less catalytic efficiency. Our kinetic characterization was in agreement with the previous work by O'Neill and co-workers, where they determined that UDP glucose, TDP glucose, and UDP galactose were the sugar nucleotides present in the highest concentration in Euglena gracilis. We were also able to determine the cellular location of the enzyme in the cytosol with a clear concentration at the vascular region. Our findings agreed with the previous works that demonstrated high levels of glycosid transferases in flagella. We further wanted to evaluate the importance of UDP glucose pyrophosphorylase and UDP glucose in paramylon accumulation. We used techniques of a single knockdown of the transcript coding for the enzyme to determine its functional relevance. From day 0 to day 28 after knockdown, every 7 days, aliquots of cells were taken to detect the enzyme by Western blot, measure UDP glucose pyrophosphorylase activity, and evaluate paramylon accumulation. Here you can see how the concentration of UDP glucose pyrophosphorylase decreases until disappear 21 days post knockdown. The mutant cells reverted their phenotype after 21 days due to the transient condition of the knockdown techniques. Here it is important to know that organisms of the genus Trypanosoma and Leishmania, also belonging to phylum Euglenozoa, have UDP sugar pyrophosphorylase an enzyme that could provide UDP glucose. We identify a nucleotide sequence coding for a putative UDP sugar pyrophosphorylase in Euglena gracilis. So we decided to perform also a single knockdown of this gene coding for this UDP sugar pyrophosphorylase and combined knockdown of the transcript coding for both proteins to determine the functional relevance of both enzymes in the paramyelin accumulation. Very quickly, the paramyelin accumulation was reduced in all experiments of knockdown, being this reduction more significant in the double silencing of the transcript coding for both pyrophosphorylases. Our hypothesis is that a fraction of UDP glucose already depleted might be redirected to compensate their levels of UDP galactose, probably through the Litoral pathway, an essential biological mechanism for the interconversion of galactosyl and glucosyl derivatives. Our work now is aimed to complete the study of the mechanism for carbohydrate partitioning and its importance for the synthesis of paramyelin. We are working on the kinetic characterization of recombinant UDP sugar pyrophosphorylase and the cloning of transcripts coding for putative GAL-T and GAL-E enzymes. We also want to characterize enzymes of the paramyelin degradation pathway, endo and exoglucanases, and phosphorylases. We are interested in knowing how Euglena degrades these polysaccharides so efficiently in vivo. We were working in the characterization of the physicochemical structure and kinetic properties of the phosphorylase described 2 years ago by Quadron Lab and co-workers. Marichal described in Grudstradt from Euglena 2 different phosphorylase activities. He called them laminaribiose phosphorylase and oligoglucan phosphorylase. We would like to identify and characterize that phosphorylase with greater efficiency working with substrates of bigger chain length. In this sense, we could identify a nucleotide sequence that codes for a polypeptide with 70% identity with phosphorylase GA149. We are trying to clone it and characterize the recombinant enzyme. I want finally to thank CONICET, National Agency for the Promotion of Science, and Universidad Nacional del Litoral for the financial support, and all people working in this project. Once again, thanks to the organizers for this great idea, which is letting us to interact with experts in Euglena's research.